all containing your gene of interest. And what you have to do now is called a plasmid isolation, which means to break open those bacteria and get your get the plasmid out of it, so you can begin the sequencing of the gene. And that's what today's experiment is all about. We just, for time and expense considerations, we're not going to be able to get radioactive probes and screen the library on the plates. We skip past that step. But what we have done is gone to the part where someone grew up one of your bacteria, one of your colonies, and today you're going to isolate the plasmid from those colonies. So as your starting material, you're going to start off with a test tube full of bacteria, and inside those bacteria are the plasmid with your gene of interest. And at the end of today's procedure, in other words, what your goal is to have a tube that contains just the plasmid. We want to get the plasmid out of the bacteria and purify the plasmid DNA from all the other molecules and organelles and stuff that the bacteria has. And so here's an overview of the procedure you're going to do today. You're going to get some microcentrifuge tubes. Your tubes that you'll be using are over here, I think. Yeah, here they are. You'll start off with a couple of yellow tubes. And then you're going to come up here and transfer some of the bacterial from the colony that's been grown up here into these yellow tubes. And the first thing you're going to do is centrifuge it. Let's see if I can get this thing open. Are you open? Ah, that's so cool. Let's see now. So there in black is the rotor, the part that's going to spin around. And I believe it'll tell you in the directions today that each group is going to do 2 tubes simultaneously. So anyway, you guys have 2 yellow tubes and you guys have 2 yellow tubes. And so you could be opposite yourself, because remember, one of the rules is you always have to have tubes opposite each other on the rotor. So that way you won't have to necessarily consult with another group to have their tube opposite yours. Everybody's doing 2 tubes. And remember that the tubes are supposed to be balanced with each other. For today, you don't have to use the electronic balance, as long as you visually see that you're adding equal volumes to each tube. That will be balanced enough. But if this was a higher speed centrifuge, you would actually have to use an electronic balance to make sure that your 2 tubes were the same weight before you spun them. All right, so anyway, you're going to load up these tubes with the bacterial culture and spin them down, which is going to pellet the bacterial cells. Now, you're actually going to do several centrifugations today. Let me give you a proper procedure here. Notice that these microcentrifuge tubes have a little hinge. I'm not sure how good your eyes are, but there's a little hinge on this side right here where the cap can open and close, like that. It's a good idea whenever you centrifuge to keep the hinge to the outside of the rotor. And the reason for that is it'll tend to pellet your whatever it is that you're pelleting down towards that particular side. If the hinge is to the outside, the pellet will form a little bit to the outside. And so it just gives you a little bit more knowledge of where to look for the pellet. Whereas if you don't make sure that the hinge is to the outside, the pellet might be anywhere towards the bottom, and sometimes for a small pellet, you won't know where to look for it. Okay, so if this is the hinge right here, and you've centrifuged, you'll get a pellet of bacterial cells. And there is the supernatant. Now, we're obviously after the bacterial cells here because they contain our plasmid. We want to get rid of the supernatant. To get rid of the supernatant, you essentially just dump it out into these little biohazard bags that you have right here. Now, don't worry about the pellet. You'll just turn it upside down and shake them like that. Don't worry about the pellets falling out. The pellet is very highly compacted down to the bottom, and it's not about to fall out. So just go like that to get as much of the supernatant off as possible. But what happens, so now we've gotten rid of the supernatant. What happens, however, is typically some of the supernatant clings to the walls a little bit, even after you shake it out. And it'd be nice to get rid of that also. And the trick for that is just simply spin the tube a second time very briefly. It doesn't have to be very long, but put your tubes in there and spin it for around 5 or 10 seconds. And that will cause any of the supernatant that's been clinging to the walls to come down more towards the bottom like that. And then you can use your micropipette to remove it. You just open this up and then holding it up so you can see it very well, stick the disposable tip down there, and suck out that extra supernatant that you spun down to the bottom. And so you should end up with more or less just pure pellet at that point, which is what you're after. You're trying to get rid of the supernatant. Sound good so far? All right, so now you have your bacterial cells. The next thing you're going to do is resuspend them in something called get buffer. To resuspend the pellet means to break it up and homogenize it so it goes back into, so the cells start floating around in the liquid again. You don't have to know what the get buffer stands for, but it's basically the ingredients: glucose, something called EDTA, and something called tris are what the components are there. And it will tell you to add the get buffer to the pellet, like that. And the trick for resuspending the pellet is this. Get a fresh pipette tip, and then using the tip of the pipette tip, dig at the pellet a little bit while simultaneously slowly pipetting up and down like that. And between the digging action and slowly pulling the pellet in and out of the pipette tip, you will break it up. You will homogenize it and resuspend it. The only thing I caution you about is this. Don't go while you do it, because that will cause the bacterial cells to start foaming up. Like you probably know, if you take milk and shake it up, it gets all foamy and stuff like that. And you don't want to foam it. So dig and slowly pipette up and down to resuspend. It could take a few minutes, but you will eventually get it resuspended. All right, so you've resuspended it. And that means all those bacterial cells are now floating around separate from each other. The next step is to lyse them open, because they have our plasmid inside, and we got to get that plasmid out. You're going to add some SDS. We use that in the genomic DNA isolation. It's a detergent, it's a soap, so it essentially breaks up lipids like the phospholipid membranes. And sodium hydroxide, which can also break apart lipids. As a matter of fact, You might remember we used that in the soap making lab at the beginning of the semester. If this is a triglyceride, and obviously I'm leaving a few atoms out here. If this is a triglyceride, you might remember what sodium hydroxide does. It detaches all the fatty acids from the glycerol. Like that. And when this happens to the phospholipids of the bacterial membrane, when it breaks apart their phospholipids, obviously that's going to disrupt the membrane. So between the actions of the SDS detergent and the sodium hydroxide base, you break open all those bacterial membranes. So what are you going to get? Well, it's going to be kind of a sludgy material at this point. There'll be some plasmid in there from all those bacteria. There'll be some broken open organelles, some junk. And mixed in with those organelles, large molecules, such as proteins. Also floating around there, along with the plasmid, are smaller molecules. Like what? Well, the bacteria probably have some glucose inside them, just because that's an energy source. Various monomers like amino acids and fatty acids and individual nucleotides, all sorts of small molecules. Salts are probably floating around in there also. You've, I don't know, almost like emptying a garbage can. You've broken open all the bacteria and all their stuff inside just comes spilling out. But at least you've at least freed your plasmid from the inside of the bacteria. The next thing you're going to add is something called potassium acetate. And potassium acetate does a couple of things. The acetate part neutralizes the sodium hydroxide. Sodium hydroxide is a very strong base, and the very high pH that we've created could potentially interfere with some of the next steps. But maybe the most important part of the potassium ions, what they do is precipitate the large molecules and organelles. In the presence of potassium ions, big molecules like proteins, the bacterial chromosome, which is a large molecule. Large molecules tend to form precipitates, but smaller molecules like the plasmid and monomers and things like that are not precipitated by potassium ions. So anyway, these guys are still dissolved, but these guys are now precipitated. And to separate the two, we do another centrifugation step. And again, let me tell you to be sure and keep the hinge to the outside when you spin. And what you'll get down here, precipitated down at the bottom, is the stuff you don't want, the large molecules. But here in the supernatant is your plasmid and smaller molecules. Well, at this step, you're not after the pellet, you're after the supernatant. And so the procedure is going to have you transfer the supernatant to a fresh tube. You can use the micropipette for that. You'll go inside there, and don't suck up any of the pellet down at the bottom. Just suck up the supernatant liquid, and then you can transfer it to a fresh, clean microcentrifuge tube. Well, we're not quite home free yet, but at least we're getting close. We now at least have the plasmid separated from the larger molecules, like that. But we still have the smaller molecules we want to separate it from. Well, the next step is to remember something we did from the genomic DNA isolation. In alcohol, in cold alcohol, DNA will precipitate, but other molecules tend not to precipitate. So you're now going to add some, I think we're going to use 95%, so almost pure alcohol. And as I said, this should cause the DNA to precipitate, but not the others molecules. So you're going to then do another centrifugation step. And now the plasmid DNA is going to be the pellet. And those other small molecules will be up there. Well, now you're going to get rid of the supernatant and keep the plasmid. Now at this point, the pellet is going to be pretty small, because if you notice, we've been eliminating more and more molecules as we go across. And so this is when it's really critical to have the hinge to the outside, because you may not even be able to see your pellet at this point. And so you just have to know that it's down